What's up, guys? Let's start with part 3, the nucleus. So the nucleus is the largest organelle and it can have more than one nucleus. So there's certain terminology to describe it. If there's one nucleus, we say that it's uninucleate, and there's a bunch of cells in the human body that are going to have one nucleus. Some specific examples in which there is more than one nuclei. So we say that it is multinucleate, and one example is going to be in the skeletal muscle. An example of an anucleate cell is going to be the red blood cells. So these red blood cells, they function as these large hemoglobin sacs to transport oxygen to these different cells. So now let's look at some of the different features of the nucleus. So we can see that the nucleus includes the nuclear envelope, the nucleolus, as well as chromatin. So what are some of those structures here? This is going to represent the nucleus, and this area here, that's going to be the nuclear envelope. So that inner layer of the nuclear envelope, that is what's known as the nuclear lamina. All of these are these nuclear pores found within the nucleus, and what it's going to do, it's going to enable the movement of those different mRNA transcripts to get from the nucleus into the cytoplasm. More specifically, when we look at this structure, we can see all of these different ribosomes studded on the rough ER where translation can occur. Nucleoli. So this nucleoli, this is the spot and where ribosomal assembly is going to occur. And then packed here within the nucleus, this is where we'll have our chromatin. So chromatin is composed of DNA. And if we look at this structure here to the right on this side, how is DNA able to go between the form of chromatin versus chromosomes? You can see the DNA, you can see the double-stranded DNA, and then you can also see what's known as these histone proteins. DNA is going to wrap around these histones, and what will form is what's known as a nucleosome. So whenever DNA is in that chromatin form, it's going to be uncoiled. In order for transcription to occur, we're able to make these transcripts whenever it's loosely coiled, as opposed to in the form of chromosomes. This is that condensed format here. Once we start talking about the cell cycle, DNA is going to have different forms here. So for instance, during interphase, so the cell cycle is divided into interphase as well as the M phase, or the mitotic phase. And DNA replication, that's going to occur during the S phase of interphase. So you have G1 during interphase, S phase, or the synthetic, and then you also have G2 before it gets into the M phase. And mitosis, so remember PMAT, prophase, metaphase, anaphase, telophase, and then cytokinesis is that's going to occur here. With M phase, that is the division of the nucleus, or mitosis is the division of the nucleus, and then cytokinesis is the division of the cell membrane. And then you can see one of the things we talked about was actin, right? Actin forming that cleavage furrow to get our two identical diploid cells. What phase is this right here, where all of these different chromosomes are lining in the middle? So that is metaphase, where they all line up in the middle. These certain different checkpoints here. First one you've got is that G1 checkpoint here, and you also have the checkpoint over here. This is going to be the G2/M checkpoint. So they're going to function to make sure we have enough cell organelles, everything is kept in check in order for it to go from one part of interphase into the next. When would we actually go through mitosis? Like, what type of cells are going to be using mitosis quite a bit? One is going to be within skin cells, right? We constantly are shedding some of these skin cells, and so they have to be replenished. Another thing is to think about is within the GI tract. So we have to replenish all these damaged cells and replace them with new ones. DNA replication is going to occur here during the S phase. So how does that process occur? The difference between the 5' versus the 3' end of DNA. So DNA is anti-parallel, and if you look here at this diagram, you'll see that this is going to be the 5' end, where we've got that phosphate attached to the deoxyribose sugar, And then the 3' end is whenever we have, this is the 3' carbon, right? And then this is the 5' carbon. We have that OH group. That's the 3' end. So you can see it in one direction, 5' to 3', traveling here, and then on the opposite strand, it's 5' to 3'. So how does that relate to DNA replication? There's two different strands. The first one here, this one is going to be the leading strand, and then this part is going to be the lagging strand. DNA polymerase is going to be the enzyme that's going to add all of these different nucleotides once we separate DNA apart. It synthesizes it in the 5' to 3' direction. So that's here in this leading strand. So that would make this parental strand, this is going to be the 3' end of the parental strand. And then this is going to be the 5' end of the parental strand. So this is that newly synthesized DNA that we're making. And so if we follow this one here, that 3' end, this is going to be our 5' strand. So this will be our 5' strand, and then this will be the 3' end, because once again, we synthesize it in the 5' to the 3' direction. That's how DNA polymerase works. It is unidirectional. So it's unidirectional, only synthesizing the strand in this direction here. And before we get to this point of starting to add these different nucleotides, remember we have that complementary base pairing where adenine will bind with thymine, and then cytosine is going to bind with guanine. In order for that to happen, one of the first things that we need is what's known as DNA helicase. So DNA helicase is going to break all these different hydrogen bonds here to separate those strands. So once it starts to separate those strands, then you get the formation of our replication fork. So this here, this is that replication fork, and then this is the replication bubble that's here on this side. With that lagging strand, so these different nucleotides, they can only be added in fragments. And so they get the formation of these Okazaki fragments, and then we have an enzyme which is known as DNA ligase is going to function to link these phosphodiester bonds. So you can see the phosphodiester bond right here connecting them together. Why do we say that it is semi-conservative replication? Well, that is because we get one new strand, right? So this strand right here, that is our new strand, and then this is our old strand. This was the parental strand that we used in order to make it. So new and old. And then we can say that the same thing on this side. So this is our old, and then on this side, this is where we get our new strand.